the major functions of amino acids and proteins. Alright. And we will take just a little bit, the very basic of other molecules, because you do study them in other modules. Okay. When we talk about molecular biology, when somebody says molecular biology, these are only some of the terms that comes into mind. Protein three-dimensional structure, DNA, RNA interaction, genes, genetic code, cloning, and all of these things, okay? All right, so today we get introduced to the molecules of life. Water is the solvent of life. We also have ions. We have energy molecules like ATP, adenosine triphosphate, GTP, and some other energy molecules. These are energy transport molecules. We have small biomolecules, simple sugars like glucose, amino acids, fatty acids, nucleotides. These are the ones that when they are joined together, they make the macromolecules. Macro means large, the large molecules. DNA, RNA, proteins, complex carbohydrates, fatty lipids, complex lipids, and so on. Okay. So to understand how macromolecules, particularly macromolecules, are made, we need to understand the chemical bonds that bond them. Covalent bonds are those bonds which are strong, bondy behasen. Why? Because they are resulting from the sharing of electrons in the outer shell of an atom, or maybe you know it as an orbital. Shell ya orbital ania أو مداراني كإلكترون تاع الصورة دوري nucleus atomica. Okay. So in biological systems, all the small molecules themselves are made as a result of covalent bonds. So for example, glucose, the bonds in glucose are covalent bonds. This is true also for amino acids and other small molecules. But in addition, we also have bonds between the small molecules to make the macromolecules. So when amino acids join together, they join via peptide bond. When they join together to make short peptides or to make polypeptides, i.e. proteins, they do it via peptide bonds, and peptide bonds are covalent bonds. So they are strong bonds and not easily broken. We have another bond in proteins' three-dimensional structure, which is disulfide bond. It is formed between two cysteine amino acids. A little more about disulfide bond you will take next week with Dr. Govand. We also have the bond in DNA and RNA. Strand of RNA, and each single strand of DNA is made by a covalent bond called phosphodiester bond. It's between the phosphate and sugar. You know, they are made of nucleotides, and nucleotides are composed of a nitrogen base, a sugar, and a phosphate. A little more about that. In details, you will take it with Dr. Haujin, the DNA and RNA structure. Glycosidic bonds, it's the bond that joins simple sugar together to make complex carbohydrates. So when glucose are joined together, for example, to make cellulose, this is done via glycosidic bond, which is a covalent bond, and finally ester bonds in lipids. So, as I said, it's by sharing of electrons. The distance between two atoms, when it's such a small distance, it causes the electrons to be shared, and once this sharing is done, Once this sharing is done, it makes this bond. This example here is CH4 methane gas. Okay, this is an example, but of course, in addition to the other examples I've just explained. Another type of bond is hydrogen bond. Hydrogen bond is weaker than covalent bonds. Okay, but it's extremely important in biological systems. It is the bond that connects water molecules together. And it is also the bond that connects the two strands of DNA together. They can be easily broken and easily formed. And this is, again, an extremely important thing, as I'm going to explain to you. Or, in a hydrogen bond, there is always a hydrogen involved. يعني hydrogen bond لا يكون ionic hydrogen هي. لا يكون دايم ionic hydrogen هي. Because hydrogen is positive, of course it has to interact with an electronegative ion on the other side. I'll show you in a minute. Ionic bonds are those bonds that are made between ionic ions, basically positive and negative ions. They exist in protein structures, for example, between positive and negative amino acids. There are also two other bonds which are the weakest type of bonds, which are called hydrophobic and van der Waals. I'll explain them, but these are the weakest type of biological bonds. Okay. So when you look at this here, please stop talking. Okay. The bond between water molecules is hydrogen bond. Within one water molecule, كwater molecule كخوي درست بي أو بيوتريب covalent چون كراسة hydrogen هي بلام چون كزور نسيكنا covalent بلام لنيواني قردكاني أو hydrogen bond. Each water molecule can make 4 hydrogen bonds, and this is quite a large number, and it gives water very special properties. Another example is in proteins. In proteins, you have hydrogen bonds that make the secondary structure level of proteins, as Dr. Govind will explain next week to you. So we have a hydrogen in one side, bond to an oxygen, usually. So hydrogen bonds exist also in proteins. And this is hydrogen bonds forming between the nitrogen bases of DNA. So the two strands of DNA are actually bonded together via hydrogen bond. Okay. بيز أحمد بو آخر جاب بيدنگ من أجر تركيز كان لقلهم نبي ناتواني بباشي طوابي ناتواني بباشي شريكم ناتواني بكاتي خويا طوابن باشا أجر برسياري كيشي كي بلين أتوان درستان برسكنا برسياري كان تاني شطوان بنفسنا وبدأ باش okay ionic bonds, for example, between sodium and chloride ions. Here sodium has an electron in the outer shell, and chloride has 7 electrons in the outer shell, so it is not yet saturated because the outer shell takes 8. So this electron moves from sodium to chloride, and this process causes this attraction, so they bond together. When they are all in crystalline form, this is how they are in molecular level. So you have a chloride, a sodium bond together, and they are bond to other chlorides, to other sodium to make this crystal. Anyway, ionic bonds exist in proteins as well, as I'm going to show you. Okay, before I go to proteins, We have van der Waals to understand. I'll explain hydrophobic in a minute as well. So what happens is that we know that electrons move in orbitals or shells around the nucleus of an atom. The nucleus being composed of protons and neutrons. Yeah, I hope you remember that from school. Right. But this movement is not circular, it's actually ellipsoid. يعني أنجولا نوي إلكترونا لدوري نيوكليسي أتومكا بشيوازيكي بازمينية. بشيوازيكي هيلكية بابلين. باشا ellipsoid. As you can see here, this is how they move. As a result of that, every atom, even if this atom is not hydrogen, even if it's not ionized, this atom will have a little bit of polarity. A little polarity means jamsardar, means that having negative and positiveness on it. It's very little, but as of this little polarity of every atom, even if not ionized, it means there will be a little bit of attraction between atoms, no matter what type they are. Okay? But because it's little, van der Waals is a type of a weak interaction, weak link. Okay. This line in purple, this represents a protein chain, a diagram of a protein chain. The protein chain itself is being made by peptide bond. Amino acids join together by peptide bond to make polypeptide chain. However, to make the polypeptide chain fold, بوي أو زنجيري بروتينة كدروس تبي لبروسيك ترانسليشن قد يخوي بقريه شوازي خوي دروس به شكلي خوي كوتاي شكلي دروس به شكل بكراوي نامني توا شن بونديك أو بروتينة واليكن أو كدي خوي بها لتوا هي وتري أو بونداني كبروتين لثري dimension structure هي لتوا. What are they? They are hydrogen bonds on the secondary structure level. They are hydrophobic interactions. What is hydrophobic interactions? In this case, for example, some amino acids are hydrophobic. They are less soluble in water. I'm going to explain which ones are they. Yeah, they are less soluble in water, that's correct. So what do? In an environment of water, they tend to get away from water and get closer together. So there is some little interaction and attraction between them. It's also a weak link. We also have disulfide bonds between two cysteines. As you can see, it's folding the protein. And we have ionic bond or salt bridges also called. Okay? All right. I'm not going to talk much about cell ions because you do take that in other modules in details and how they act, especially in second year module, membrane nanoreceptors, which I'm going to be with you in that module as well. So, sorry, before we go to ions, let's talk about the properties of water. I'm going to talk about ions in a minute, sorry. So water is a solution of life. As you know, a large percentage of the cell ناتوام نمن تركيزكم نطلب كان تركيزكم كيشتان شيء بيدن بن برسياري كيا كيشية كيا من أبقى عيم زور بضمنية بوب حاضرة بلام أمدان بالا باشا أجر جوتان لينية ياشتي نازانم نايبين اللي بشتوا كيشتان يتكايا بيدن بن. In the cell, a large percentage is water. We're talking about mammal cells, eukaryotic cell, even prokaryotic cell. But in human, there are some exceptions, like bone cells, for example. But we also have water outside the cell, in between the cells in the tissues, interstitial fluid, and we have plasma as well. Water is essential for removing waste from the cell and from the body. Is essential in keeping the temperature of the cell stable and of the body. But it's most importantly, no biochemical interaction can occur without water, because all molecules, they keep their structures and they interact together through water. Water is everywhere. Why is that? Why is water on a molecular level, what is very special about water? Well, it's polarity. It's a polar molecule, very highly polar molecule, because it's composed of oxygen atom and 2 hydrogen atoms, positive and negative. But in addition, you can see from the other image, so you can see from the other image here, is that it has a bend structure. It has an angle. It's not a flat molecule. It's not composed of oxygen and 2 hydrogen like this. If it was flat, it would not have been very polar. So it's not just the component of water. It's not only the component of water, it's the bending, the shape of water molecule. So this bending is very important. Why? It puts the oxygen mostly on one side and the 2 hydrogen mostly on another side. As a result of that, water can make 4 hydrogen bonds, each water molecule, and it's very polar. Why is it polar? Means jamsadar, you know. Why is it important? Because we will see from the structure of other molecules, either biomolecules, that most of them are either charged or polar. So they need a solution, a medium, which is polar, to be able to deal with them. As a result of their polarity, other molecules, either charged or polar or non-polar, are either hydrophilic, they dissolve in water, they interact with water, or they are hydrophobic, like lipids. They are water-hating, they don't interact with water. And there are some molecules that are amphipathic. Amphipathic is when molecules have both properties. Let's take some examples. So you can see glucose. Glucose here is composed of many atoms, but it has many hydroxyl groups. Hydroxyl OH, hydroxyl group. Hydroxyl group is minus, negatively charged, and that's why all these groups, it dissolves in water. This is true for other simple sugars as well. Fructose or even the sucrose composed of 2 sugar units and so on. Glycine is an amino acid, the simplest. Aspartic acid is another amino acid. Again, they have many charged or polar groups. Lactic acid, glycerol, and so on. When it comes to lipids, for example here, this is wax. Okay? Yes, there is a C-double-O. This is a carboxyl group, and carboxyl groups are negatively charged. But this is a very small group. This is just one group compared to what? If you look here, look what is written. CH2 by 6, CH2 by 7 here as well. معناه شيء، معناه شش CH2 هي. بكرتي أولك ليرة how CH2 يتر هي. معناه شيء. These are long hydrocarbon chains. They are not charged, not polar. They don't dissolve in water. This means that this molecule will not dissolve in water. Cholesterol, for example, cholesterol has an OH group. Yep, OH group here. structure cholesterol. بلام هر أويا هموز أو دواي structure كي زوري structure كي ناتويت ولا أو نبرة ناتويت ولا أو فرم.
Is that structure composition
No. These are not. You can ask. It's good to ask this question. Thanks for asking. But sometimes also, if for example you forget to ask such question, use your common sense. I would not want you to learn this by heart. All I want you is to recognize the groups. أبي بون منا بزانن hydroxyl group chia, carboxyl chia, hydrogen atom, proton chia, phosphate group chia. أمانة biological groups هي PO4 minus. زور دي تبردم كان. إذا باسي أمينو جروبا كاين لا بروتينا لا أمينو أسيد كانا أمانة ما دير لقلي الناس know. Anyway, here let's look at amphipathic. Phenylalanine is an amino acid. It's almost, you see, it has two groups charged, but also has a non-charged group, non-polar, doesn't dissolve in water. A better example, phospholipids. In this example is phosphocholine, phosphatidylcholine. This is the component of cell membranes. It's composed of this. It has fatty acid chains. Fatty acid chains, these are long, than CH2 by 15. Water. Fatty acid chains, but they also have large phosphate head groups composed of PO4, phosphate, composed of glycerol, and composed of the choline part as well. Okay. Now, when a molecule has almost 50%, almost maybe 40-60% of two groups in its structure, in that case it becomes amphipathic. تقريبا نيوية توز زيادة توز كمتر للستركشر كي charge bipolar بيبتويت ولا أو دوينتويت أو PO3 amphipathic. This is really important for membrane structures because the phosphate heads in membrane, plasma membrane, are projected out and in of the cell. Out of the cell, in of the cell, what do we have? We have water. And in between we have the fatty acids to make the insulation. Persiartabo?
Phenylalanine and lactic acid. Both of them have, one of them has carboxyl group and one of them has carboxyl group and O. What the difference between them? One of them is hydrophobic and one of them is
This one and this one?
yes.
Yeah, our structure is آخر ليرة سنة ليرة أمبرومة هذه يكفي البرومة أما هيدروجينا الليرة أما هيدروجينا تروحنا أمبرومة دورة دوانا أما هي جروب ECH3 بلام ليرة سيريكا. You see here, this is a very large. This is CH2, CH3, CH2. أما aromatic usually non-dissolving in water except if they have something like OH. For example, in lysine. بدل بالبيز أحمد. Alright? As a result of water polarity, water dissolves ion, and water can dissolve ion faster and in more quantities than any other solvent because it has something called dielectric constant. It's very high. It simply means that it can dissolve ionic compounds much in a larger quantity and faster. So once anything is dissolved in water, like you can see in this here, in this diagram, it means the ions are separated from each other by water molecules. I will come back to this in a minute. The second property of water is that it's composed of hydrogen bonds. Each water molecule can make 4 hydrogen bonds. They associate together. Water molecules associate together by hydrogen bonds, and they associate with other molecules also by hydrogen bonds. As a result of the hydrogen bond of water, water becomes a little, if you like, sticky. Actually, water has 2 properties called cohesive and adhesive. نزل بكردي شيء أمانة خلوا الدنيا گردەکانی ئاو تێکەوە بە پاڵاتی ئەنووسن بە هایدرۆجین بۆند واتوانن بنووسێن بە دیواری مەسەن تیوبی ڕەگی ڕووەکەوە adhesive ئەوە cohesive and adhesive. So basically they are very sticky, okay? This hydrogen bond, this stickiness gives water the property of having a high boiling point. So until you get to 100 degrees Celsius, water doesn't boil. All right. Also, it gives water the property of high surface tension. High surface tension is because of this stickiness, because of this cohesiveness. And high surface tension causes water to do the capillary action. Capillary action is the movement of water against gravity up in very narrow tubing. This is how water moves from the soil into the roots of a plant and through the roots to other parts of plants. It is capillary action. It is because of hydrogen bonding of water. Also, the movement of water in and out of ion channels and special water channels in and out of the cell is also due to this high surface tension. In addition, it is also because of the hydrogen bonding that water, when it is frozen, ice has less density than liquid water. I'll explain this later at the end if there is time, because you're not required to go into the details. Okay, so as I said, water can move rapidly in and out of the cell through channels, and it can take materials with them in and out of the cell. Okay. So when we say something is dissolved in water, we mean that every molecule of that material is surrounded by water molecule. So if you look into the diagram of sodium chloride here, you will see that sodium ion is separated from chloride ions by water molecules. Now, if sodium chloride, if salt, is not dissolved in water, here, as you can see from the lower image, sodium chloride will be clustered together, all the ions will be clustered together. This is undissolved. But once they are dissolved, every single ion will be surrounded by water molecule. This is true for anything that is dissolved in water. So glucose, for example, or sucrose or whatever, once dissolved in water, it means that every single molecule of glucose or any other sugar, simple sugar, is going to be surrounded, existing close together, yes, but they are surrounded by water molecules. This is the meaning of dissolving. Of course, the larger the molecule, the larger the number of water molecules required to surround each molecule. This is common sense. This is a true three-dimensional structure of a protein. All the little parts you see around it, أو شتى بتشوفاني لدوري هموي آون، قردي آون، أو قوري خوي structure كراستي protein. It's the three-dimensional structure of a protein, and this is only colored. You use the special computer programs to color it, but it's actually the true structure. So only to know this, understand the meaning of dissolved. Something dissolved in water. Cell ions, as I said, I'm not going to go into details. You know that we have potassium and sodium. Especially potassium, we have it more inside the cell than outside. Sodium is the opposite way. We also have calcium, chloride, magnesium. We have bicarbonate, phosphate, amino acids, proteins, because they have charges. Some amino acids and proteins made of them. They are also ionized materials. And the difference in concentration of these ions between the in and out of the cell is extremely important for cell signaling and for controlling the osmosis regulation of the cell and so on. But this is enough because you take it in other modules. Okay. So let's come to pH and buffers. We need to understand this quite a lot because we will see that pH affects the structure of amino acids and proteins, and also that amino acids and proteins are themselves are buffers. First of all, we need to understand acidic, basic, and neutral. So solutions and materials are either acidic or basic or neutral, as you know. This is depending on what? On the concentration of hydrogen ion. So instead of measuring the concentration of hydrogen ions, you know, in mathematics, when there are some values that they are really large, they are in large quantities, mathematically it is easier and more practical to use the logarithmic value. So that is why pH is the logarithmic value of hydrogen ion concentration, but it's a minus, pH equals a minus logarithmic value of hydrogen ion concentration. As a result, any solution pH is 7, you know it's neutral. Any solution which is below 7 is acidic. It means it has a higher concentration of hydrogen ion. It's the opposite. Higher than 7 is basic, and it has a lower concentration of hydrogen ions. And of course, when it is more in hydrogen, it's the opposite with hydroxide. Okay. Most biological solutions are neutral. For example, blood, the pH of blood is 7.4, the pH of cells inside cells, cytocells 7.2. And it's all biological fluids. You need to know it. You can't be doctors not knowing the pH of blood, cytosol, or cerebrospinal fluid, and so on. You already know most of this. Just look, for example, gastric acid, it's about 1 or less than 1. Please stop talking. بين ختم سمكة تكايا إذن. Okay? Gastric acid is very acidic. You know that. Yeah, usually normal people know that. You don't need to come to university to know that. It's 1 or less than 1. It's very acidic. Why? Because of the existence of HCl. In this diagram, you don't need to learn this diagram by heart. أنا please ناكل بالكن. أنا بس معلومات خدتها. أما بس خشتك بالي. أما تنهاي بيشانية دالنا ويك خانة راسها بقشتي حوت بوينت دوا pH كا. بلا لبشكاني خانة كم يجاوزي هي. وان ونا لايسوسو مكان جوريك لأورجانيل وكو قدي خانة سيرة كرين the stomach of the cell. Because a lot of digestion process happen in it, lysosomes are a little acidic. Okay? And there is a little bit difference in mitochondria and so on. But generally, you need to know that the cytosol, the cell, pH of the cell is 7.2. Right. But the problem is, in the cell and outside the cell, in blood, in between, in the space between the cells, there are continuous biochemical interactions, continual biological processes, which means that the pH is bound to change. If the pH changes, it affects the structure and therefore the function of all biological molecules. They will not be able to function. So how is the pH kept stable? It is kept by solutions or materials known as buffers. Okay, so to understand what is a buffer, let's remind ourselves what is an acid and a base on a molecular level. On a biochemistry level, an acid is any substance that can donate a proton or an H plus ion. Proton, not protein. Yeah, hydrogen ion, yeah, proton. A base is on the other side, is any substance that can accept a proton. Okay? شو هالشيء H plus يهبه بزوري هيبه أو أسيدا أو بيتبس باش. Many of the components of the biomolecules in the cell, in biological systems, they act or they have compounds that act as acids or base. يعني يا خو يان بقشتي acid base يان بشري كان acid يا base. And as of their interaction, acids and bases are made as well. Okay, so let's look into this equation part. Don't worry too much about the equation. I will tell you which part I am trying to get to. So first of all, a general equation of acid and base. For example, if you have an acid here, HA يرى acidic, acidic قشتي. ماشي؟ Could be HCl, كاليرة Cl, could be H2SO4, كاليرة SO4, and so on. When it is in an aqueous solution, the environment of body, of cell, of blood, whatever, it will be ionized. There will be hydrogen ions or hydronium ion. Don't worry too much about H3O. Basically means the H plus will join the water. And there will be the A minus, which is the conjugate base. Okay. For a reaction like this in chemistry, we have something called dissociation constant. What does it mean? It means that we want to know the rate of this dissociation. أولى يجيبوا نوعي أم أسيدا شنيك خيرايكي نسبتكي شنيك راشكي لا environmentكي لا أو الآلي. So we measure the dissociation. Dissociation means delinking, يعني جيابو نوا. Dissociation constant, okay. What we are concerned with is acid dissociation constant, Ka, which shows how much an acid can dissociate or can lose a proton. Protonic If we look into this original interaction up there, the HA with H2O, our constant, our كي لدر كان ليرج دي سنة H2O كي لدر كان H كمان يتوا هربا بسيطي HA بيتا H plus A minus معناه شيء معناه هر أسيدية كجيا بيتوا كديسوسييت بيت. What do you do? How do you measure the dissociation constant? You take the resultant, the H plus the A minus concentration divided by HA, which is the original acid before dissociation. باشا أما بس بوي بيشان ماندا أو أسيدا شنيك آمادية أو جابو نويلة به شنيك آمادية ionized به بو أما جرينجا. طبعا أوا بس بوي بزانن أي minus كابيو تري conjugate base. Conjugate base شيء هر acidic H plus كاني ليلادا أوي أمانية توا conjugate base. باشا منا ومننا وختاي منية أوي ناينا وهناينا باشا. Don't get confused by it. For example, HCl, when you move the H plus, what is remains the L أو كاتبيو تري conjugate base. باشا. باشا حاول يعني أول كامل كتب باشا. Okay, and HA is of course the concentration of the acid. Okay, فضل.
Okay, كبو rate dissociation هي بو quantity amount
آخر ما اشتكى rate يرى أو معناه يأذى. باش. Why is this important? Stop talking. بيدنك بين. مناقشات لبيني خوتانا. من استكم جورتنا اختي زياد قرمة. وايا؟ لا. ماشي دي من برا صار كم خمسة. alright ماشي هو أنا عشت Why is this important? Well, because based on the dissociation constant, acids are either strong acids or weak acids. This is also true for bases or alkalines. Bases are either strong or weak. But we concentrate on acids more because in biological systems we have mostly weak acids. باشا بوم مهمة weak acid base strong acid base إنك الشيء علاقة مع هي اسم بس يكي. First of all, we need to understand all the molecules of a strong acid, once dissociation occurs, they dissociate all of it. So for example, HCl, H2SO4, HNO3, nitric acid. These are strong acids. Means what? It means once they are dissociated in an aqueous solution, in a solution basically, what happens? All the molecules of that material will be dissociated. همو قرديكي HCl كا بوتا H plus Cl بجيا. باشا؟ وواتاي چی ديتوا؟ واتاي او ديتوا لو solution لو جينجا چی زورا H plus زورا. H plus sorbate, it's very acidic. But when we come to a weak acid, of course I said HCl, a weak acid, no. Not all of the molecules of that acid will be dissociated. Acetic acid, for example, amino acids, by the way, proteins, because they are made of amino acids. Yes, some amino acids, I'll explain why amino acids in a minute. Amino acids are also considered weak acids. Weak bases as well. Okay, I'll explain that in a minute. But acetic acid exists in biological system. Bicarbonic acid exists in blood. Phosphate buffer also is composed of an weak acid. These are all exist in biological systems. It means acetic acid, for example, once dissociation, not all of the molecules of the acetic acid will be dissociated into H plus and the conjugate base. Perhaps maybe part of it, maybe 40%, maybe 50% only will be dissociated. So in a solution, maybe half or less or a little more of this acetic acid will remain in this form. The other half of the molecules will be in this form, the conjugate base and the H plus. This is called the weak acid. Okay. Why is it important that we understand weak acids? I'll come to this in a minute. But before that, it's easier to not measure the dissociation Ka, dissociation constant directly. It's easier, just like the pH, it's easier to consider the pKa value, which is the logarithmic value of dissociation constant. So a pKa for any acid, for any material, is defined as the pH أو pH 50% of dissociation occurs. Okay? As a result of using, of course it's minus the logarithmic, so as a result of using a logarithmic value, exactly, becomes like the pH becomes the opposite. So weak acids have high pKa, strong acids have low. Alright? As I said, there are also weak bases and strong bases. And the majority of medicines, in addition to the importance in biological systems, أو درماناني ودروس تكرين أسبرينا باراسيتامول زولا درمانانا زور بي بشيوازي دروس تكرين الفارماسيوتيكال كومباني شركاتي درمان كدروستي أكن بشي ريسيرشكا بويك أسيد دروس تكرين. لبردو هوكاري سركي هوكاريكي أويا بو أوي لبي بلادو لبي خانه خانه كان بأساني interaction كان ور بگیرترین هوکارێکی تری ئەوە لە سڵە دیسێشنەکان ئینشاڵڵا پرسیارتان لێ ئەکرێت جوابێدەنەوە باش. Okay, so when you have a neutral solution, if you add acid to it, it becomes acidic. If you add base to it, alkaline to it, it becomes basic. Okay? So this is why we care about understanding weak acids, particularly weak bases as well, but weak acids because this is what exists in biological systems. Buffer solutions are what resist the change in pH. buffer solution كان أو ماتريالاني كبفرن أوانن كناهيلن pH خانة زور بقورة لأنجاني biochemical أو همو تشالاكياني كلنا وخانة ولا وبيك هاتوا لشي لweak acid and of course the conjugate base. So that is why weak acids are important because they make buffers. Each buffer has what is called a buffering capacity. So if you look here, this is the buffering titration curve for acetic acid, also called ethanoic acid. Okay? What does it mean? It's simple. You will have this بينك بالبيز أحمد. أما هي titration curve هي بوهريك لأمينو أسيد كان بوهرشتي كweak acid بين. weak base شبه. شيء titration كا؟ أما تجربيك أكري تحقيق النوعية أكري بزانن كPH زياد كلا هنا pH بله pH أم أسيدا كهيدروكسالي تكرا الكلايني تكرا دروب باشا دروب باي دروب تكرا بهنا كي تايبت أو كي تايبت لمختبرة أم أسيدا شنيك pH كي أقدره أميريك همويك أسيدك لمنطقيك لناوچيك تايبت بو أسيدا زور ناقدره pH كي واتا خيرائي قراني pH كي كمتر أو كمتر أقدره pH كي استليرا لنيواني 3.6 acetic acid has a buffering capacity. واتا نايل pH كي زور برزوا بين كهيدروكساليتي أكي. تواناي بفري هيلة. بلام بيش أوا بينا شون برزبيتها ودواي أويش بخير أي برزبيتها. أما طبعاً بو أمينو أسيد كانيش أو لا سبوديسيش مكان أمينو أسيد كم بوتان هنا وتوا. titration curve كي همان فكرة بزانن لكيو أم أمينو أسيدا أو كي pH يعني buffering capacity زيادة وشون أقولنا. So this is the area shaded in blue is the buffering region of any weak acid, and in this region there will be 50 in the middle of it. So if you look here. In the middle of it where it says pKa, ليرة pKa now راستي أو شوني كبشين شيء كراوة أويا pKa لو كانت شيء أبي أسيدك لسطع بنجاي ديسوسيت ولا سطع بنجاي ديسوسيت نبوه. Okay. Buffers usually have only a capability of resisting pH in their pKa plus and minus 1. يعني هر بفريق بفريقين تابون هنا بفريقين حوت بوينت شوارع حوت بوينت شوار بينج حوت بوينت شوار بلين سيب هين حوت بوينت 35 حوت بوينت شوار 5 أو أنا تقريبا توزي زيادة كم ترلوا أتواني بفريقين بهيلتوا بس stable أو يوم بس تكا. واتا اگر اسیدی کی زور ترچه یا بیسی کی زور نتوانیم مقاومه بس لو حدودا خوشی لو سنوره بس. Okay. This equation, this relationship between pH and pKa is known as in this equation known as the Handerson-Hasselbald equation. pH equals pKa plus the logarithmic value of the concentration of the conjugate base and the divided by the concentration of the acid. Biological buffers, I've already told you, the phosphate buffer in cells, bicarbonic buffer system in blood, the amino acids and proteins themselves. Here, this is basically this diagram. It just shows what it says there. Phosphate buffer here. Phosphate buffer here. Carbonic buffer, yeah, bicarbonic buffer here in the blood outside the cell. لإنسايد السيل فوسفيتد هي بروتين وأمينو أسيد كان لدروا لجوروا هردوكي بس ليرا أول إضافة كرو اللي أجر RBC تبي خروج السورة كان ليرا هيموغلوبين دوري كي تايبتي هي أم جدولة أم خشتيا بو إيوه أبي بيزانا نعلم همو شيء لبر كانوا بيزانا تيايا نا كيسناك بلام أبي بيزانا مش كإيوه من الطبيب إن شاء الله أما باسي أسيدوس باسي شار حالتك blood acidosis respiratory acidosis metabolic acidosis respiratory alkalosis metabolic alkalosis وتا كPH لش زيادة كازور يا كما كازور حالتك چيا چون تريتمنت تكريم بلي وكشتيكي قشتي بلي مطلوبة ليتان بيزانا أما ليتان دواء لهمو بزور بلي موديولا كاني تريش Alright, amino acids تنوي بردوام بين يا بريكي كي كرت ورقرين أنا بريكيويا أنا هاتوا عادة چیاله؟ بریک ورگره؟ باشه بس بریک که بعد دقیقه اگر بس عمرو کیتوا جم سردار چیه؟ پولار چیه جم سردار؟ اگر کسی کزانیه ولام داتوا پولار یعنی چی جم سردار؟ شکی کر بالبتیه جم سردار بیدن بگه زحمت بس فرق باشا طواوا اگر بشوازي تركسي تربيله بلي واتا بلي راسا هردوكتا واتا هل قديك كبيبو تريجم سردار بولار واتا او قدا چي تيايا هم بوزيتيفي تيايا هم نيجاتيفي تيايا وعادة لا يا بوزيتيف ونيجاتيف كي تقريبا ايكوال تقريبا لبرا وبيا وتريب بولار Okay, باشا that is polar باشا فرسياري ترك أكرا عليه فرسياري تنشكرا عليه أمل بركين من فرسيار وانا ينمو بلهم والله هالشي تخطيطي هين هيا بوم بنوسا فرسياري كي راستا وخوف هكسي بينش لبركة تواني لبريكا فرسيار بوش شوية ناعتوا إن شاء الله حفتي دهاتو ستاديسيشني أم بابتا ورقرن بەوە فکرێ وەرەگرن لەسەر پرسیارەکان بەڵام زیاتر پێش ئیمتیحان نمونەی سامپڵی پرسیارتان پێدێ بێدەنگ بن سامپڵی پرسیارتان پێدێ و سامپڵی وەڵام پێش ئیمتیحان لە کۆتایی ئەوە زیاتر فکرەیەکتان پێدە بەڵام ئەبێ بیزانن ئەم شتانە یعنی ئەمە ئەبێ بزانن یعنی من ڕەنگە پرسیار بێنمەوە بڵێم بۆ نمونە لە مالتیپل چۆیس بڵێم ئەو بەفەری وا بەرپرسە لە the buffer which is responsible for keeping the buffering of the blood is one bicarbonic buffer, phosphate buffer, other buffers. So of course you know mostly it's the bicarbonic buffer. Anyway, let's come back to amino acids. The second part of the lecture, I hope it's going to be shorter and simpler, hopefully. So amino acids, of course, you know, are the basic units of building proteins. And we will see why proteins are extremely important. Okay. They make short peptides, amino acids, and they also exist as single amino acids. What amino acid تنها لبضية لهمو منوركان تنها بوانية بروتين جروبس كان خويا وكو single amino acid يشان إسا إبينين وكو short peptide يشان جيجالا protein. So amino acids can act as either an acid or a base, and usually, well, always, not usually, always, they are, when they act as acids or bases or both, they are weak acids, weak bases. So amino acids are affected by themselves by the change of buffer. But they are also themselves have buffering capacity. هلا بر أول لو diagram بيشتاني دا in the diagram it shows that not only bicarbonic buffer and all the other buffers, but also the proteins are made of amino acids, and the amino acids as part of proteins as single amino acids, they also participate in buffering the cell because they are weak acids, weak bases. Alright, some amino acids are very soluble in water, others are non-soluble. Amino acids that exist in proteins are in the L form. There is the D form and the L form. Those are the L form. Here is the general structure of an amino acid. بزحمة أو قراني أو لا بتشنا مجالة في تعمل قصة زوركن. بيدين. So you have the carbon alpha, carbon inawant, carbon alpha peutre. We have the carboxyl group, COOH, the NH2 basic group, the H and the R. Arakachia kiazani, arakachia lera. Variety. Wata araka hamu amino acida kan, hamu au bashani ter wakuyaka, hawawati wakuyaka. بس لأركيا لو شريك نسراو أرك جاوازة. جروبا كي بيري جاوازة أو ما أكا أمينو أسيدة كبقورة. In physiological pH, the pH is normal of blood or of cell, they exist in their ionic form, and it's called the Zwitter ionic form. Zwitter ionic form is where they are dipolar. Boom, because the carboxyl has lost its H, and the amino group NH3 has gained an NH. As a result, it is C-double-O minus, this is the carboxyl group, and NH3 plus, this is the amino group. Okay, this is the Zwitter ionic form of an amino acid. Okay, this is the general, again, just shown the general structure of an amino acid. The carbon in the middle is carbon alpha, C-double-O, and NH3. The lower image here, image shows an amino acid in the three-dimensional structure. So it's not flat, this is all it has a three-dimensional structure. Here are some examples. You don't need to learn the structures by heart, but the 20 standard amino acids here, the names. بلام structure كان هنا مهرواسي كان بوي أشنا بالمهدي structure كان. So methionine and cysteine are two amino acids that have sulfur بزرد كراوليرا in their structures. So they have a three-dimensional structure. Some of them are small, like glycine. Some of them are large, like tryptophan. And some of them are in the middle. So amino acids, because they are the same in all parts of their structures except the R group, they are classified based on the R group, obviously. Most commonly, this is done based on charge and polarity. So charged amino acid, positively charged, negatively charged, polar amino acids that have both charges on them, negative and positive, and those which are non-polar. Of course, sometimes other classification is also mixed with this. We have three amino acids which are aromatic, and the rest are aliphatic. Aromatic, what they have ring structure. Okay. You are not required to learn the structures by heart. Structure كان كيستا كان لبري كان بلام أبي أشنا باللقالي because يعني زور شد هي بيتي بقى الستركشر كان. أما نيرا طبعا نسراوا نانبولر أم جماعة نانبولر جلايسين لهندي لكلاسيفيكيشن كان نيرا داندري بس جلايسين أكل لهوي داندري. جلايسين are group KH. Atwan, not polar or polar, don't be confused by it. I would put it with the polar one. Okay? I would put it there, so don't worry about this. Those parts which are shaded by pink, Ambashana, are the R groups. So we have alanine, glycine is the smallest, it's very important. Glycine, أو amino acidae, و زور لهم protein بروتينك تقريبا نسبة جلايسيني هبه بلام لو بروتيناني وبيستي بفلكسبيليتي هي لا جوليا زياتر لا إشي بروتينا كان راجي جلايسين زياتر أشكا أمينو أسيد بتشوكا مجال بحركة ده anyway alanine, proline, valine, methionine, isoleucine and leucine. Okay, so this is the first group. We are now talking about the 20 standard amino acids. Standard amino acid account 20. Again, أول شيء ده now it was confusing. Don't worry about it. ألين polar and charge. ما بس تيجيها. خوي كويت polar براستي أمل عادي. ما بستكي أزانيشيا ما بستكي أويا كما ما دام هدو تشارجك هي لسر آر جروبك طبعا ما يسكى عليه بولر ونكو ما بس ما ما بس ما آر جروبك هي شو كأجر بس أمينو أسيدك ما قشتي بكين همو أمينو أسيد كاربوكسيل جروبك هي و NH3 هي okay. So all amino acids we have the bullseye. We are talking now on the R group. So if the R group contains both charges, it's polar. ما بس دي aware charge كي يكتري cancel. Okay, we have those 5. Serine, threonine, cysteine that has sulfur as well as methionine of course. Asparagine NE طوابع glutamine. چونکه دوانی ترمان هاي ناوكي لواكا بلام they are negatively charged. Okay, so we have the positively charged amino acids. They are 3. Say positively charged man here. Histidine, lysine, arginine. We have aspartic acid and glutamic acid as the negatively charged. And we have the 3 aromatic tryptophan, tyrosine, and phenylalanine. Again, no, you're not required to learn the structures by heart, but you are required to know the names of the 20 standard amino acids. So amino acids have the acid and base property. This is due to the carboxyl and the amino group, but also if there are any charged group or polar group in the R group. So, depending on the carboxyl and the amino group and what is existing in the R group. So, I'll explain the second sentence here on the diagram. Amino acid that lack ionizable R group exist as weaker ion. This is shown here. Come this diagram. So this is important because like any other acid or base, amino acids can be protonated or deprotonated. So they can gain protons or donate protons. ما بس أويا proton بدأ يا proton ورقة protonated or deprotonated. It will affect the behavior of the amino acid. It will affect the behavior of a protein and structure and function of a protein. Some amino acids as part of a protein, which is an enzyme, by being protonated or deprotonated, this is how they do its enzymatic function. Okay. So in this diagram here, okay, all this means is this, that زرد تعقيدي ما كان لخوتان بس أولية. All this means is that this is a general amino acid. Normally, it has a carboxyl COO-, NH3+. This is the amino group. Basha? Normally, an amino acid exists regardless of the R group. Of course, if we consider the R group, this will change a little bit. So, what happens is that this is the sweeter ionic form. If the amino acid exists in an acidic environment, it will be protonated. وطبعاً هنا نرى H زيادة لا carboxyl group ما بيوتريم protonated. احتمال H زيادة شتا R group بجوري جوري R group ماشي؟ Right. If it exists in an alkaline area, in a basic area, it will be the opposite. It will be, see here, it is NH2. It has lost a proton. Okay? So basically, this just shows this. All I want you to understand from this diagram is the fact that pH, the environment pH, will protonate or deprotonate the amino acid, and as a result the protein, and it will affect the structure and the function of that amino acid. Okay? Amino acids, as I told you at the beginning, they associate together by peptide bonds. They bind by peptide bond, which is a covalent bond. It is formed between the carboxyl of one amino acid and the nitrogen of the amino group of another amino acid, as you can see in the diagram. So you can see here كربوني كاربوكسيل amino acid يكم لقل نيتروجيني amino group amino acid دهان. A molecule of water is lost and a peptide bond is formed. It's a strong bond, it's not easily broken. These are the names. The 20 standard amino acids. بله أبي بيزلنا يعني بلا محاولة بكن براس توخو أو هلا بري كان توخو تان هلا بري أكن أو أنا ديتا بردم تان نمونة تان في أدري أما جورن كيوايا ما إيشا كيوايا هرب الأبري أكن بس أبي بيان ناس نوا بو أوين ستاندرد كان بناس نوا بو جي بيانو تري ستاندرد for only one reason كاين غلطة لك هي غلطك حتى عندي ما مستيقظ يبو أو غلطة كان ألين piano three standard لبروي بشكل structure protein. This is a big mistake, a very big mistake. باشا أو يبغى إعداميكم. They are called standard only for one reason and one reason only, and that is that their codes exist on genes. إما لسر جينا كان شيء من هي كودون من هي. كودون من هي. كودون شيء بونونا ATA, ATC or whatever. nucleotide ويا كودوني كودي أمينو أسيديكا. ونيا أو أزانا أو يدويا كودوني أمينو أسيديكي ترى. أو كودونا تنحو بس أمينو أسيدك. أم بس أمينو أسيديكا بيانو تري ستاندرد. وتأيما نان ستاندرد إيش من هي؟ نان ستاندرد كودون يعني لسر أمينو لسر جينكا. كودون يعني لسر جينكا. بلعن هر خويا لجوران كاري standard amino acid كان درستنا. So modification of standard amino acids is what results in non-standard amino acids. I have given you some examples here. For example, collagen, which your skin is composed of it mostly. Your cornea, the cornea of the eye, in tendons, even in the structure of bones tissue, you have collagen. Collagen has many amino acids in it. Two types of amino acids that are non-standard, hydroxyproline and hydroxylysine, they exist in large quantities in collagen. Hydroxyproline are made from proline hydroxylated, an additional hydroxyl group added to it. The same thing for lysine. Two enzymes, do enzyme كأن إيشاء أو برولینا كنا هيدروكسيبرولين أو أنا لبروتينا كدواء بروتين درست به أو إنزيمانا ببي فيتامين سي كارناكا هذا بر أويا أو تري بوبيست فيتامين سي جريكا أما بس بويش لكان رأيته gamma carboxy glutamic acid exists in blood clotting proteins. Dopamine is a neurotransmitter, I'm sure you know it. Dopamine is a non-standard amino acid. But we have something else in amino acids. In the 20 standard amino acids, half of them are essential, the other half are non-essential. This is only relevant to human. أما بس علاقة بإنسان وهي. Why? Because half of those amino acids, our bodies can make them. The other half, we have to get it from the diet, from food. Our bodies, human bodies, only cannot make them. And I'm talking about the synthesis of the amino acid itself, not the synthesis of proteins. باسی دروستکردنی ئەمینۆ ئەسیدەکە خۆی ئەەکەم چۆن دروستە بێت. نیوەی ئەوانە ئەو بیستە نیوەی نەناو لەشمان دروستە بێت نیوەکەی تری ئەبێ لە خواردن ئیسمانکرێت. ئەمەیە ئیسێنشەڵ و نەئیسێنشەڵ جیایە لە ستاندەرد و نەستاندەرد. Functions also, you know that they are part of proteins, of course, but they also function as single amino acids as in communications between cells. Some hormones like thyroxine are amino acids. Histamine is also in paracrine signal, it's also cosmic hormone, is amino acid. Neurotransmitters are mostly either short peptides or amino acids. Glycine is neurotransmitter, dopamine as well. They are important for metabolic reactions. For example, most importantly, nucleotides that are the units for DNA and RNA, they are made actually from amino acids. The nitrogen base is made from amino acids. So, citrulline and ornithine are two non-standard amino acids in urea biosynthesis. أو دو amino acid لشوتيا زورا. هلا بروا عليها هم بقولشي للباشا. In urea biosynthesis, urea metabolism. Okay. And some in plants they have protective functions. We also have peptides, dipeptides, short peptides, oligopeptides, and they are involved in signaling and they have hormonal function. We have some examples here: oxytocin, glucagon, that prevents blood glucose level from dropping too much, glutathione, and so on. These are only few examples. You will take more examples, I expect, in metabolism and other modules. Okay, so let's come to proteins. Why are proteins important? So proteins are the functioning molecules of all organisms. بنزحمة تركيز ملقلاب كان أول شيء كمان سواء سنة زور. أثاني زور طالبين بدن بن. Okay. So هرشي functioning ليش هي همو بنورك لهمو خانيك همو بنورك بروتين وتنها بروتينيك. All the other molecules are either energy molecules or they are source of information, genetic information. But when we say a gene is expressed, gene expression means the synthesis of protein. Gene ka khoy hich naka, DNA ka tanha bashya kalam zanyari. Paragraph ek waragre RNA ka messenger RNA ka beta paragraph la DNA copy khoy kurdwa. ودرس كدني بروتين فانكشن كاكا هرشي فانكشن هي تنها بروتينيكا حالتي زور كم هي RNA هندي جوري RNA وكم وكو انزيم كاركا همو بروسكاني خانة دروي خانة همو بروسكاني living system بروتينيكا. They also make structures of the body. Yes. انزيم تقريبا لسطانة والتوبينجي زياتري انزيم چيا بروتينا أوي كأمينة ولا انزيمات چيا RNA
يعني بس أول شيء الانزيمات
نكبشة هل خوي انزيم بروتينا أو سديك أسأل هل انزيم بروتينا أو نكبشة يعني أول شيء كأليم RNA انزيم كان خوي RNA خلني بلام انزيم كان انزيم هي باشا بلام زوريني انزيم بروتينا. Okay. They also make part of the structure of the body. I'm going to give you examples. So as I said, proteins are the products of gene expression. When a gene expresses itself, it actually makes a protein. Our traits are either proteins or they are the result of protein reaction. همو صفات كانمان بينراو ولا بينراو رنگي قجمان بالامان رنگي چالمان شكلي دم چالمان لناو انزيم كانمان همو اوانا اما صفات جيني همو ان صفاتانا همو يا بروتينا يا انجامي بروتينا ونمنا ميلانين رنگي چاو اواكا رنگي بيس رنگي قجمانا ميلانين لانجامي انزيمك بيوتر تاروسينيز دروستبه او دو ان شاء الله نمنيك but everything about you, structurally, your eye, your skin, your hair, everything about you is made of protein, and every single function in your body and in all creatures is made by proteins. We'll take some examples. So they are composed, usually proteins are either 50 amino acids or more long in chain. Okay? They are composed of 20 standard or also non-standard amino acids. كل هالقرآن كاري بس هني لنا standard كان دروس به أو بيتا non-standard. Anyway, so there will be non-standard amino acids as well in protein structures. The amino acids in general, they work as alphabets. وكل شو alphabet هي بابلين أجر هاد بيستكا حساب كين ستاندرد كا الفابت يكت هي بيك هاتوا لبيس بيت لو بيتانا أتواني شن وشدروس كي زور إناش بيرتي همان شتا لبر أوا بروتيني زور دروس تبي لوي بوشيواني كأمينو أسيد كان يكتر لوي بروتين دروس. Each protein as a result will have a special molecular weight. وزني خوي بيو تربا دالتون يا كيلو دالتون حساب أكريم. And this great diversity in function is due to diversity in structure. It can do all of the different functions because it can have different structures, even though it's all made of amino acids. And to understand protein function, you must understand protein structure. Before we go to the functions, something very important. Every protein, because it's made of amino acids, and amino acids act as bases on acids, weak acids with bases. So all proteins will have their own particular pH, PO3PI, isoelectric point. The isoelectric point, this is the pH. When the pH is at a specific isoelectric point of that protein, the net charge will be zero. أجا protein يكمان هبه PI كي pH به ل pH pH دايني precipitate هبه چونك charge نابه. أو زيادة ل study session كان مناقشة كين لبرسيارة كان وباسية كين فاشا. Okay. Basic protein will be positively charged. Acidic proteins will be negatively charged. Basic proteins like what? Like, any idea where do they exist? Our proteinane combine DNA kala chromosome. Histone proteins. Okay. All right. So what are the functions of proteins? Well, everything. Enzymes are proteins. An example is lysozyme, but there are many. Transport and storage. Hemoglobin transports materials, and it's a protein. Ferritin stores iron, it's a protein. Mechanical force, actinomyosin, they are proteins, but not only actin and myosin, microtubules as well. They are important in many functions, including division of chromosomes. هر كشيء كبين microtubule كان chromosome كان بيكساني جانا بنوع لكاتي مايتوسيس وأبيته كاري يكلنا نخوشية أو كان نخوشية جنتك كان. For example, Down syndrome. Anyway, so all of these are fiber proteins, fibrous proteins. Protection, antibodies, immunoglobulins, or protein. Providing strength and structure, collagen, elastin, keratin, amana hamwi protein. Receptors. All receptors that are involved in cell-cell communication, sending signal, receiving signals, all receptors are proteins. Opsin للشاوة هيا لشبكية شاوة هيا رودوكسين بوشو بينيني شو أوبسين بينيني روجا. It's a receptor, receives light, and it causes vision process to happen. Hormones, some hormones, not all, are proteins like insulin. Transport across cell membrane. Ion channels, carrier proteins, كواكن أو شكاني في استبجت ناو دروي خانة. All of these are proteins. And adhesion proteins are those proteins that connect cells together in a tissue, and they have many functions. This here is an image of some proteins. I'm only showing you this as examples just to know how much diversity they are. You will see other examples. من اگر زوزو بلن بزحمت بدن من نتوام زو طابه بدن من تركيز كان. Finally, just a little bit بدن. أم جارب ببینم ده کسی قصاکا ببره لأن داوكم قاعكا بجيبه بدن من. DNA and RNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. DNA is the source of genetic information in all cellular life, basically in all life systems. Viruses are an exception. They are not considered exactly live anyway. So we have three major types of RNA: messenger RNA, transfer RNA, ribosomal RNA. All of them are involved in protein synthesis. Okay, you will take these in details with Dr. Haushin, but you also have other types of RNA. Some are enzymatic, some have other functions. DNA and RNA are made of nucleotide, and each nucleotide is made of a nitrogen base. Base nitrogeni, چونکه nitrogen زوره structure كان amino acid كان درست كده بأساس. And the sugar and a phosphate group, PO4 minus group. Okay, they are each of these linked. Many of these linked to make either RNA or DNA, and that is because of the existence of PO4 minus a lot. Both RNA and DNA are acidic molecules. You know RNA differs from DNA. RNA is one-stranded. RNA has ribose sugar, not deoxyribose sugar. And it has uracil instead of thiamine as a nitrogen base. Carbohydrates, they are energy sources for the cell. They also provide structure for some organisms. For example, cellulose is made, plant cell wall structure is made of, it's a complex carbohydrate. We have monosaccharides, one unit of sugar. We have disaccharide, oligo, and polysaccharides. And monosaccharides could be ring shape or linear, depending on how many carbons it is. And glucose, is made from photosynthesis, in photosynthesis process, and it's broken down to produce ATP as a source of energy. The bond is glycosidic bond, as I explained earlier. Examples of disaccharides is sucrose, lactose. Oligosaccharides exist in biological system as part of glycoproteins. زنجيري كورتي carbohydrate بروتين أو بسرعة glycoprotein درستها يان بلبيدوا beta glycolipid, and they have very important role in immunological processes. Polysaccharides like cellulose, glycogen, chitin, starch. Lipids are hydrophobic molecules, and that is why they are the perfect molecule to make cell membranes. They are long-term energy. They storage energy. There are fats and oils and phospholipids, steroid hormones are lipids. So hormones can be amino acids, can be short peptides, can be proteins, can be lipids. Okay, we have saturated and unsaturated lipid. Saturated when there is no double bond in the structure. أجر double bond دي هبة أو unsaturated double bond للstructure. أجر نيبة أو saturated أو الدواي لممبرين أو receptors لاسيتين أو بوتشيكرينجا. It is composed of fatty acids and glycerol. The major part that we type of fat in human body are the triglycerides. Jorakani triglyceride كبير كاتونا fatty acids and glycerol. أو structure ولا بالكردنيا بس بوتيجيشتنا كشون triglycerides درسنا. I am finished for today. أويد برسياري هي أتواني ببينك بس بيزحمت بينك إيش ولا شيء خوتاني بيننا وأويد برسياري هي أتواني دور.